Hello friends, welcome back to my YouTube channel Journey with Vijay Kumar Shrivastav. Today I am going to present the topic Cotton Hybrid Seed Production Technology. Before presentation I would like to introduce myself. I am Vijay Kumar Shrivastav. I have completed M.Sc Agriculture with specialization in Agronomy from Pant University of Agriculture and Technology, Pantnagar in 1996. So let's move to the presentation. Now we will discuss about cotton crop. Cotton is a major fiber crop of global significance. It is grown in tropical as well as subtropical regions of more than 80 countries. The major cotton producing countries are China, India, USA, Pakistan, Uzbekistan, Turkey, Brazil, Greece, Argentina, Australia, and Egypt, which are contributing about 85% of global production. India has the largest area under cotton cultivation in the world, approximately 9.1 million hectare, and grows all four cultivated species in cotton like Gossypium hirsutum, Gossypium barbadense, Gossypium herbaceum and Gossypium arboreum on commercial scale. About which, related to hybrid seed production, generally Gossypium hirsutum and Gossypium barbadense are used. The major cotton growing states are Maharashtra, Gujarat, Andhra Pradesh, Tamil Nadu, Karnataka, Punjab, Haryana, Rajasthan, and Madhya Pradesh in India. However, the productivity of cotton in India is low due to its rain-fed cultivation in major areas and inadequate use of quality inputs. So in textile industry, cotton is playing major role for the supply of industrial requirement. As the importance of cotton, there are various usages like lint. This is the main part of cotton crop, which are used in textile mills, thread industry, yarns for various kinds of fabric making, medical cotton and mixing with synthetic fiber. After that, second use is cotton seed oil. Cotton seed contains nearly 19 to 22% of oil content, which is used for cooking purpose and mostly used in Gujarat. After that, cotton seed meal cake, which are used for livestock feeding and organic manure. Then linters are there, which are having the high grade writing paper use. Then photographic paper and X-ray films preparation. Then stocks are also used, which are in pulp and paper industry, manufacturing of hardboards and corrugated boxes. So there are various uses of cotton. As green revolution was major milestone in Indian seed industry for food grain production, like same white gold revolution is related to cotton crop. Farmers planted Bt cotton seeds on 95% of cotton cultivable area after its introduction. India's production doubled and India became the world's second exporter and producer. India's Bt cotton success is a result of partnership between farmers, seed and biotech companies, agriculture universities, and trades and governments and non-government organizations and news media. As we are going to study about hybrid seed production technology in cotton, so first we should know what is hybrid seed. Hybrid seed is the seed which is F1 generation produced from a cross between genetically dissimilar parents, and the hybrid vigor or heterosis is the absolute phenomenon where expression of the characteristics such as growth related traits, flowering behavior and yield fall outside the range of its parent with superiority. So in case of hybrid seed, the yield data and its performance is better compared to its parents. In hybrid seed production, the seed is the main input and the seed source should be pure and it should be of highest standards. The foundation seed should have quality as per seed standards and it should be from truthful sources. While if we are going for hybrid seed production, we have to use two parents in hybrid cotton seed production, which is known as female parent A and male parent restorer line R line. In hybrid seed production of some crops like maize, pearl millet, sorghum, rice, mustard, sunflower, castor, sorghum Sudan grass, the female parent A line is sterile parent. While in case of cotton, okra, chili and tomato, we are following emasculation procedure to make female sterile because originally the female parent is bisexual flower which contains both male and female organs in its flower. So we have to go for emasculation process. The points which are important in cotton hybrid seed production, that first, A line female parents should have desirable characteristics, male parent R line having desirable characteristics, and hybrid F1 should having also desirable characteristics as per the requirement. In cotton, there are two methods are used for hybrid seed production. First one is conventional hand emasculation and pollination method, and second, non-conventional, where the male sterility system is used. Conventional method is majorly adopted in all production areas for cotton hybrid seed production, and non-conventional method is less adopted. In case of conventional method, it is highly labor oriented and expensive, while in case of non-conventional, there is less labor requirement and it is less expensive compared to conventional method. So slowly the production system is moving from conventional to GMS or male sterility system based. First we will study about conventional method of hybrid seed production in cotton. Majority of hybrids released in our country are of conventional methods. Development of such hybrids involve three steps. First one, identification and growing of male and female parents. Second, emasculation of female parent, and third, pollination of female parent with identified male parent. Cotton is an often cross-pollinated crop and the average outcrossing is about 6%. The pollen is heavy and sticky, so air movement is limited, and the cross-pollination occurs mainly by insects like honeybees and bumblebees. Hybrids have been released for commercial cultivation in Gossypium hirsutum and Gossypium barbadense species at tetraploid level. And these hybrids are of two types, like intraspecific and interspecific. Interspecific means hybrids which are crossed between two different species, and intraspecific means hybrids released with crossing between same species. In diploid cottons like Gossypium arboreum and Gossypium herbaceum, conventional method is highly uneconomical because the ball setting is low due to a small flower size and brittle pedicels. Second method of cotton hybrid seed production is male sterility based method. The development of hybrids using male sterility eliminates the process of emasculation, since the anthers are sterile in female parent without pollen, because of which the cost of hybrid seed production can be reduced. However, the pollination method is same like conventional system. In cotton, mainly two types of male sterility is used. First one, genetic male sterility, and second, cytoplasmic genetic male sterility. In cotton hybrid seed production, generally genetic male sterility system GMS is preferred. Cotton flower is having bisexual nature, means same flower contains male portion like stigma and anthers, and having the female organ also stigma. Here in female, in GMS method, the parents are having 50% population of male fertility and 50% male sterile plants. So rogging is compulsory to remove male fertile plants before crossing starts or pollination starts. Here we have shown the pictorial representation of crop and process stages in cotton. It starts from sowing, followed by germination, then seedling, then vegetative stage, then a square formation stage, which is also known as pre-flowering stage. After that, flowering stage, which is having the two processes like emasculation and pollination, then ball development stage and bursting, then cotton lint picking stage. So these are the stages which are coming under field stage. After that, plant processing stage starts from ginning, then delinting, after that mechanical cleaning, after that manual hand picking and sampling process, after that dispatch of final produce, and onwards testing and results. So these are the stages of processing and testing stages. Here we have shown the pictures of field stages in hybrid seed production, and after that processing activities where the implements are used. We will develop in broad about each stage and processes. Now we will proceed through cotton hybrid seed production principles and practices. First one is field selection. In field selection, we have to select medium to heavy deep soils which are well drained and considered ideal for cotton seed production. The fertile, well leveled with good irrigation and drainage system fields should be selected where same crop was not grown in previous season to make free from volunteer plants to avoid admixture and any quality issues related to genetic as well as physical purity. In field preparation, the field should be fertile and well prepared for good germination and good crop growth. Now seed rate. What is seed rate? The number or quantity of seeds planted per unit of area per acre or hectare to ensure optimum plant population for maximum yield is called seed rate. In case of conventional hybrid seed production, the female parent seed rate is 480 to 600 grams per acre, while male parent seed rate is 240 to 300 grams per acre. In GMS hybrid seed production, the female parent seed rate is 1 kg to 1.5 kg per acre. In case of GMS hybrids, we are going for dense planting because there is 50% rogging. So in advance, we have to plant dense population which after rogging will come to the normal. And male parent seed rate should be 0.4 to 600 grams per acre. Seed rate may vary hybrid to hybrid depending upon seed size and parent growth nature. In cotton hybrid seed production, generally wider spacing is maintained and manual sowing is done, so seed count plays important role. Sowing time. Correct sowing window should be followed as per traditional historical databases for high yield. Mitigation measures should be followed to escape from natural hazards as rain damage during ball bursting and picking and disease and pest attacks. So, suitable window period should be preferred. And sowing method. In case of cotton hybrid seed production, generally manual or hand sowing is done. Depending on parents' idiotypes, we have to arrange row and plant spacing to maintain optimum plant population for high yielding. In general, there is a square method or rectangular method which is followed for cotton hybrid seed production. Now spacing. In cotton hybrid seed production, we are following two methods, conventional hybrid seed production and GMS hybrid seed production. And both systems, the spacing will be different. In case of conventional hybrid seed production, we have to maintain generally female spacing of 4 by 4 feet or 5 by 4 feet or 4 by 3 feet or others depending on the suitability of female parents, plant idiotypes and growth pattern of female. In male, we have to maintain abundant population for the availability of flower, for sufficient supply of pollen for pollination and crossing during whole period. The male spacing should be 3 by 2 feet or 3 by 1.5 feet or 2.5 by 1 feet depending on growth patterns. The male line is used only for flower availability for crossing purpose, so there should not be shortage during whole crossing period. In case of GMS hybrid seed production, there is approximate 50% rogging in female, so there should be optimum plant population in field. We have to maintain generally female spacing of 3.5 by 1 feet or 3 into 1.5 feet or 3.5 by 2 feet depending on plant idiotypes and growth pattern of crop female parent. In male line, we have to maintain a spacing same like conventional hybrids. Depending on parent idiotypes, we have to arrange row spacing and plant spacing to maintain optimum plant population for high yield. Planting ratio or row ratio, it refers to the sowing arrangement of rows of female and male lines. This is followed in hybrid seed production where two parents, male and female lines are used. In cotton hybrid seed production, instead of row ratio, block method system is followed where female is sown in one block and with isolation distance of 5 meters, male block is planted. The female and male block ratio is generally vary from 80 by 20 to 75 by 25. Staggering. Staggering refers differential sowing of male and female parents, means difference between sowing dates of both parents to achieve proper synchronization of flowering of both parents during flowering in hybrid seed production. Staggering depends on flowering behavior of both male and female parents. In cotton hybrid seed production, generally staggering is not required, as cotton crop is indeterminate crop where vegetative and reproductive growth continues for longer periods simultaneously. Even sometimes in male parent, a split sowing is done in 2 to 3 steps to get abundant flowering for uninterrupted pollen supply during crossing period depending on male floral behavior. The aim is to synchronize both female and male flowering. Isolation distance. There are isolation standards varying from crop to crop, and for best genetic purity it should be complied strictly. Maintenance of genetic purity for certified seed production of conventional or male sterility based hybrids depend on the use of safe isolation distance. The cotton hybrid seed production plot should have 30 meters isolation distance on all sides from other cotton fields. The isolation distance between two parents, male and female, it should be minimum 5 meters. The isolation distance within parents is followed to avoid any admixture while pollen collection or crossing or picking. So, boundary side total crop should be 30 meter isolation distance from other cotton fields, and within parents it should be 5 meters. The isolation distance vary from class to class of seed. In foundation seed production, it should be 50 meters between cotton crop. And in case of certified seed or hybrid seed production, it should be 30 meters. When the cotton crop is having the different polyploidy crop, then we can maintain the isolation distance of 5 meters in both foundation seed and certified seed, because there is no chances of contamination, and we have to maintain this only for avoiding any admixture or adulteration during field operations. While sowing, we have to take precautions. The fields should be well prepared, well pulverized, and while sowing, the male and female parents should sown separately. There should be no any mixing chances, and we have to follow the timely sowings under suitable window period. If the field is not vacant, then we can grow female and male parents as a seedling separately. The polythene bags of size 10 by 15 centimeters can be used, containing fine mixtures of soil, sand and farmyard manure in the ratio of 1 by 1 by 1. Seedlings can be grown in vacant a small part area and which can be transplanted later. The seedling can be transplanted within 15 to 20 days after sowing with optimum soil moisture. So the raising seedling is the one of the methods when we can opt for transplanting if the field is not vacant, and we can transplant the seed after the proper growth and field is available to be sown. This picture we have shown the germination process. In cotton there is epiphyseal germination where the cotyledon is coming out from the soil after germination. In this picture we have shown the germination process. Germination and early seedling development where the root growth dominates the early growth of the plant. Gap filling is one of the important process in cotton hybrid seed production. Whenever the germination is not proper in fields and the gaps are observed, then to fill the gaps we have to use the seedlings for gap filling, and the gap filling should be done only in conventional hybrid seed production system, and it should not be done in genetic male sterility system based production. For gaps we have to sow 10% of the female and male parent seedlings, and it should be done on same day of female sowing to escape late gap fillings. Gap filling of seedling should be done within 15 to 20 days after sowing in optimum soil moisture conditions, and care should be taken while transplanting and gap filling to avoid any mixing of male and female seedlings, and we have to ensure 100% female population for high yields and ensure high female plant population 2000 to 2500 plants per acre in case of conventional hybrid seed production, and 4500 to 5000 plants in GMS production system. and the optimum spacing can be followed 4 by 2.5 feet or 4 by 3 or 4 by 4 feet depending on the parent's nature. There should not be gap filling in female in GMS production system, because in genetic male sterility systems we are going for rogging, where 50% rogging should be done, and if the seedling growth is not, if the crop growth is not uniform, then rogging will be a issue, it will be a problem, and it can cause genetic purity problem. So gap filling in GMS based system is avoidable. Now to study about crop management in cotton hybrid seed production. It can be described as a agronomic management, which refers to the proper management of crop supplying fertilizers, nutrition and irrigation timely. Good agronomic management results to good yield and quality. After that, field monitoring. Scheduled field monitoring is very important for supervision of crop from sowing to harvesting stage, to ensure timely operations for best quality seed and good seed yield. After that, plant protection measures, where we have to take care of weed control, insect and disease management. Then quality management, which refers to the management of genetic and physical purity. Then post harvest management, which includes picking of cotton seed lint, then drying and storage of seed cotton lint. After that, plant processing operations, which consist of ginning, delinting and mechanical cleanings. After that, sampling and dispatches, we have to take the samples of final produce. It should be sent to quality test and dispatch of the produce to the processing plant. So we have to manage the all procedures which is involved in hybrid cotton seed production. Here we have shown the some snaps of cotton hybrid seed production fields, which refers to the management system. In case of cotton hybrid seed production, we are using mulching system also to control the weeds, and it is included with drip irrigation systems to conserve the soil moisture, and the growth will be very good in such crop and we can get the good yields. After that, we have to incorporate the organic materials, timely earthing up operations should be done and proper maintenance should be there. These are also some snaps from the cotton seed production fields, where we can see the earthing up of crops for better management and fertilizer application. If the management is good, the crop will result in a better growth and ultimately it will result in high output. Along with fertilizers, we have to supply the additional nutrient to the cotton crop in hybrid seed production. Like 60 to 65 days crop, we have to go for foliar application of NPK 19-19-19 at the rate 1 kg per acre. It will boost up the growth and development of the crop. During 70 to 75 days stage, there should be foliar application of NAA or Planofix at the rate 6 ml per acre, mixing with 10 liter water. It will result in better square formation and flowering, which will result for good crossing to get the highest number of balls and seed setting. After that, 75 to 80 days, we have to go for foliar application of NPK again, which will provide the nutrient supply to the developing balls. And during 85 to 90 days stage, again we have to go for Planofix spray at the rate of 6 ml per acre within 10 liters of water, which will again regenerate the flowering process. We can get the new squares for crossing purpose. So by giving these additional nutrients, we can regenerate the growth and flowering. Now, plant protection and nutritional spray schedule for cotton hybrid seed production. This refers disease and insect pest management timely to prevent damage to the crop, additional supply of nutrient to the crop, and appropriate steps should be taken timely for good yield and quality management. Here we have given the schedule for a spray of chemicals and additional nutrients which are needed time to time, and all the chemicals and additional nutrients are included here, and the timing and dosage are provided when it should be sprayed. It will be very helpful if you are following the schedule, and ultimately it will result in plant protection measures for pest control and additional supply of nutrients to the crop depending on the crop requirement, and it will result in better growth and yielding. Now water management. The cotton crop cannot tolerate waterlogging condition and prolonged drought also leading to soil moisture stress. Adequate drainage provisions should be there, and after monsoon rains there should be drainage facility, and earthing up should be done with blade harrow after fertilizer application to provide adequate drainage and to facilitate water application. Irrigation interval depend on the soil type and distribution of rainfall, and care should be taken to maintain adequate available soil moisture soon after fertilizer application and blooming and ball formation. Now flowering. Flowering refers to the full blooming of the reproductive parts of the plant where pollen moves from male organ to female stigma for pollination. And synchronization. It refers to the simultaneous flowering of male and female parents in hybrid seed production is known as synchronization. Synchronization in flowering between the parental lines assumes greater importance as the seed set on female parents depend on the amount of pollen supplied from the male parent during flowering period. Crops should be in frequent observations to check nicking or synchronization status for same time flowering. There may be little variations in flowering based on climatic condition and flowering behavior. So in such case, corrective measures should be taken. Male line 2 days advancement is preferable. Here we have shown the flowering of cotton where during the square formation stage there will be pin head square which develops initially. Later it's developed to match head square, then it converts to a square growth midpoint, then it will be take the shape of candle, and after that there will be blooming of the flower. Here we have shown the cross sectional structure of cotton flower. Cotton is a bisexual flower which is consisting of both male and female organs. Stigma, style, ovary and ovules are female organs, while filament and anther male organs. During emasculation process, all male organs like filament and anther is totally removed along with petals and bracts, and only female parts as stigma, style, ovule and ovary is left on the plant part. We have to maintain proper isolation distance as earlier we discussed to avoid any contamination to the quality. Crossing should be started only after complete vegetative growth, and we have to remove the flowers in early stage to get complete vegetative growth. If flowers are left on plant, because of this, if seed setting is there, ball development is there, ultimately vegetative growth will be impacted. So for certain periods we have to remove all flowers which are available on plants and let's plant to grow well. If crossing started without complete growth, development will stuck and ultimately there will be impact on flowering and ball development. Here to study about male management. Male is very important in cotton hybrid seed production as the female yield is depending upon the male supply. There should be 5 meters gap between male and female parent. Male should be planted one side in a block isolated with female, and male planting should be done in open area which is exposed well to sunlight. If well sunlight is there, there will be good flowering, good square formation and pollen availability and its bursting, because of which we can supply the pollen in sufficient quantity to the female crossing. More male plant population is needed to get abundant flower supply during whole crossing period. Don't allow ball development in male line till crossing continues. If we will allow the ball development on male plant, definitely there will be impact on flowering. If crossing continues for longer period, there will be shortage of male flower and ultimately there will be low yielding. So we have not to allow the commercial ball development on male parent during crossing period. Emasculation process. Removal of male organs, stamens or anthers or killing the pollen of a flower without damaging the female reproductive organ is known as emasculation. Emasculation is one of the important practice in cotton hybrid seed production in conventional method. In bisexual flowers, emasculation is essential to prevent of self-pollination. In some crops, like hybrid seed production of cotton, okra, chili, tomato, emasculation process is followed to make female sterile. Here we have shown the some pictures from cotton hybrid seed production fields which refers to the emasculation process. For emasculation, the suitable size of buds candle should be selected, and we have to remove the bracts and we have to remove the petals along with stamens and filament, and after that we have to put red tag for demarcation, and then crossing is done. So we can see here after emasculation the only female organ like ovary, ovule, style and stigma is present, and all male organs has been eliminated from flower. During emasculation process, certain precautions are needed. There should be selection of appropriate size of flower bud, checking for inadvertent retention of anthers during emasculation. There should not be anther particles left on stigma, avoiding damage to female parts during pollination. Emasculated buds are marked with red color polythene rings for easy identification during crossing on next stage. The best time for emasculation is 3 to 6 pm, and we have to avoid poor emasculation. There should be a strictly removal of inadvertent self-pollinated balls and open flowers. And emasculation should not be done in the morning, otherwise it will impact the quality, because in there the cotton hybrids having the different flowering natures. Some hybrids, some flowers are sensitive, in which the bursting is taking place, pollen bursting is taking place inside the bud in early hours. So we have to not follow the emasculation process in morning at all. It should be done only in evening hours. Pollination. The movement of pollen from the anthers to the female stigma by wind or by carrier or manual transfer is known as pollination. Pollination or crossing is essential for seed set and therefore crucial in seed production. The manual crossing is also followed. In case of hybrid seed production, pollen transfer from male plant anthers to female stigma manually is called manual crossing. It is followed in hybrid seed production of the crops where emasculation is done like cotton. In cotton and other crops like okra, chili, tomato, the manual crossing is done. During pollination, the emasculated buds are pollinated the next morning with the pollen of male parent. The best time for pollination is 8 to 12 am, which depends upon the temperature, because the stigma receptivity is maximum during this period of 8 to 12 am. Generally 4 to 5 buds are pollinated by one flower of male parent. Crossed balls can be identified by the absence of bracts and visibly can see formation of rings at the lower side. Crossing works are effective during the reproductive phase from 60 to 120 days. So the crossing period is about 2 months to get the maximum yield. And we have to avoid the ball development in male parent. If ball development is permitted, there will be shortage of male flowers, and ultimately there will be less crossing period which will result in less yielding. For emasculation and pollination, 8 to 10 levers are needed in conventional hybrid production for 8 hours per day, and in GMS hybrids 5 to 6 levers are needed for 4 hours for pollination per day. Here we have shown the some snaps from cotton hybrid seed production fields where the male anthers are collected and they are arranged in different way. Some where direct male flower buds are used for crossing purpose. Some where the pollen are separated and it is kept in a bowl and the stigmas are crossed with powder pollen powder, and in some cases these powders are filled in syringe and the buds are pollinated with syringe. So in different regions different methods are adopted. Here we have shown some pictures from cotton hybrid seed production fields. Here we can observe there are red rings which are kept on a stigma after emasculation, and after pollination these rings are removed. We can see here the different stages like ball development stage is there, crossing stage is there, as cotton is a indeterminate nature plant where the vegetative growth and reproductive growth are taking place simultaneously for longer period. This is also one snap from cotton seed field. After crossing, regulating the size and spread of female parent by nipping offside and top shoots is advisable. It will increase the ball size and seed weight. Giving light irrigations frequently at the time of crossing and ball development is needed. Applying potassium and sulfur fertilizers to gain seed weight. Don't allow cell balls. It's more powerful than cross balls in development, and also cell balls are threat to the genetic purity. Here we have shown the picture from seed field where the crossing has been stopped now, and now the bursting is taking place. After crossing, the nipping has been done and the apical branches has been pruned out to avoid any other vegetative growth. So now only reproductive growth will take place. Here we have shown the ball shapes of cotton seed field. The ball shapes will vary depending upon the parent nature. It can be oval, it can be conical, it can be round, or tips should be pointed. So different ball characters will be there as per female parent. There are various factors which are affecting cotton hybrid seed production like weather conditions, sowing window, agronomic and plant protection measures followed, vigor and characteristics of female parent, pollen supply potential of male parent, male and female planting ratio, a staggering pattern or synchronization, male flower to female flower crossing ratio, a stage and period of crossing, labor engagement and labor availability, ball retention capacity, post-harvest care, and plant processing operations handling. So these all factors are ultimately affecting cotton hybrid seed production. Here to study the yield attributing characteristics in hybrid seed production. Like first one is morphology of female parents. Second is monopodia and symphodia arrangement. Third is square formation and flower bearing capacity. Ball retention. It refers to the retention of crossing balls. Sometimes what happens after crossing the balls are falling down, which is the loss. So wherever the retention percentage is maximum, it will be good for yielding. After that, number of balls, size of balls, ball weight, seed size, number of seeds per ball, ginning percent, cleaning seed recovery percent, and seed test weights. So these are the factors which are playing important role for the yielding. Here we can see the morphological characteristics of female parents like its monopodia and symphodia arrangements and ball bearing capacity and ball arrangement. Under cotton hybrid seed production, there are quality parameters for testing okay. The seed produced is going through the testing like GOT grow out test results should be more than 90%. The produced seeds germination should have 75% and ELISA results should be 90%. So if the seed testing is achieving this criteria, it will be declared as pass or accepted. And where the GOT is less than 90%, germination is below 75%, and ELISA test is less than 90%, will be reflected as reject. ELISA test refers to the Bt content in seeds. Under cotton hybrid seed production, there are quality parameters for testing okay. The seed produced is going through the testing like GOT grow out test results should be more than 90%. The produced seeds germination should have 75% and ELISA result should be 90%. So if the seed testing is achieving this criteria, it will be declared as pass or accepted. And where the GOT is less than 90%, germination is below 75%, and ELISA test is less than 90%, will be reflected as reject. ELISA test refers to the Bt content in seeds. Now we will discuss about quality threats in cotton hybrid seed production. We have to remove open flowers in female parent, and there should be no self walls, and dumping of emasculated and open flowers outside, and remove unwanted flowers in female regularly to get continuous flowers. It will cause regenerations. So after removing the excess flowers, we can get new flowers in addition in few days. Rogging. Rogging is one of the important process in seed production. Rogging refers to the act of identifying and removing plants with undesirable characteristics from agriculture or seed production fields. Rogs are removed from the fields to preserve the quality of the crop being grown. Rogging should be done timely to rog out undesirable plants as off types, pollen shedders in A line, semi-sterile plants, volunteer plants, other crop plants, and disease heads, etc. For effective rogging of off types, it is necessary to have knowledge on the distinguishing characters of identification of A and R lines. And the rogging stages in cotton, it should be done timely and prior to any contamination. It is done from vegetative stage to harvesting based on the many parameters as morphological and reproductive characters. The critical stages are most important in cotton hybrid seed production as vegetative stage, pre-flowering stage, square formation stage, flowering stage, ball development stage, and maturity and picking stage. In cotton hybrid seed production fields, rogging should be done in male block first before crossing start to avoid any contamination, because during crossing we are taking pollen from male parents and crossing to female stigma. So male block should be totally 100% pure, free of any contaminants or off types or any volunteer plants. So male rogging should be done on priority basis. Rogging is compulsory in male and female blocks. We have to remove male and female mixing. Rogging should be done on the basis of plant height. It may be tall or short, depending on branching pattern, having the characters of open or erect or bushy type. Rogging parameters can be also based on leaf size, leaf color, lobes, hairiness, and glabrous natures, and basis of squares, size of squares, buds, stigma. Then after that, flower color, anthers and pollen color, petal colors, ball size and shape, arrangement of balls, and cell balls should be removed. So there are different parameters for rogging. If we are going for cotton hybrid seed production, we should have well information knowledge about parental quality regarding morphological characteristics and floral characteristics. As floral characteristics are concerned, we should have clarity about petal colors, petal spots, these anther colors, then length of the stigma, after that bract size, so on the bud size. So on the basis of this, we can identify if any contaminants are present. And we should check while picking of male flowers and emasculation, if any differences are coming, immediately we have to rog out. Before pollination, we have to also check out the male floral characteristics, like its color, stigma colors, pollen color, length of the flower. So if any differences are observed, immediately we have to sort out that male plant. Here also we have shown some characteristics to be observed while emasculation and pollination process. On regular basis, we have to remove the self balls in female line and male line also. In female, it will help to get continuous flowers for longer crossing period. and to get assured quality and to get good ball size, seed size and weight in crossed balls. While in male, by removing of balls, it will promote continuous flowering till crossing ends and it will be suitable condition for supplying of the pollen for female requirement. Here we have shown the pictures where the precautions should be taken while emasculation. There should not be any anther presence on stigma because it will contaminate and it will proceed for self-pollination. So ultimately there will be threat to quality. So while emasculation, all the filaments, anthers and pollen buds should be removed and it should be totally clean. Only a stigma and female organs should be present. Here to study about harvesting and post-harvest technology. The kapas should be collected from fully opened crossed balls and picking refers to the manual collection of seed lint from bursted balls, collecting fully burst crossed balls regular basis, and post-harvest operations. The post-harvest handling is the stage of crop production immediately following harvest, including drying, cleaning, sorting and packing. So after harvesting we have to take care all post-harvest operations and processing activities. While picking, crossed balls should be picked carefully and there should be no mixing of self balls. The identification marks in crossed balls, there will be no breaks in backside. So we can identify easily while picking. Self balls should be strictly picked separately and it should not be mixed in seed produce. We can observe easily there will be presence of breaks in balls. So it is the indication that these balls were not crossed while seed production. So we have to take care by the removal of these balls and avoid mixing with seed produce. After bursting, the immature balls should not be picked as it is the loss to the farmers. If mistakenly picked out, it should not be mixed in seed cotton lint. It will cause germination problems due to immature seeds and while testing, there will be subsequent germination which will lead to the rejection of the lot. Kapas damaged by bollworms and fungal diseases should be removed and discarded. Kapas which are free from debris like dried leaves, bracts, etc. should be picked and it should be cleaned. The picked kapas should be dried well under sun for 4 to 5 days till it attains optimum moisture levels, and kapas should be picked in afternoon hours when it is dry, and dried kapas should be stored in gunny bags under well ventilated conditions for further ginning works. Ginning is one of the process which is related to cotton crop. Mechanical separation of fudge seed or gin seed or lint seed from seed lint is called ginning. This process is followed in cotton crop produce. While ginning, we have to check machine and floor cleaning. We have to take care while bagging and there should be no any adulteration in gin house. So we have to take care because this is the produce which we are getting after long hard work and following the different processes of seed production. We have shown here the ginning operation process. The ginning machines are used in which the seed lint is fed, where the lint and seed is getting separated, and these seeds are called gin seed or lint seed, and these seed should be dried under exposure to the sun to get the optimum seed moisture. De-linting. De-linting is the process of removal of fudge from cotton gin seed. This is the chemical process where acids like sulfuric acid is used, where the concentration is 93 to 98% and it is used at the rate 100 ml per kg of gin seed. In some cases, gases are also used. In de-linting process, this gin seed is mixed with sulfuric acid in drum where there is rotating motors, and after mixing the lime water is added and it is run for approximate 3 to 5 minutes which will depends upon the quantity of seeds. So this is the chemical process where the seed is mixed with acid and it is rotated by mixing the lime water, and after that the fudge is washed out. So here we can see the pictures where the lint has been washed out and now white seed has been converted to black seed. After delinting, we can easily find out the physical purity of the seed. If there is mature seed, it will be totally black color, well filled and having the good seed size, while in case of immature seed, well, the bursting was not taken place well, it will be light colored and the lot of empty seeds will be there, so there will be higher processing losses. Here we have shown the equipments which are used in cotton seed processing, like cleaner machine, after that gravity cleaning machine, pin sorting machine which is used to separate the cut seed and pin whole seeds, and after that finally we are going for this hand picking which is done by laborers, and also we can find out sorting machines, color sorting machines, and after that bagging and storage of the seeds. Needle sorting machine is used when the crop is infested with pink bollworm problem, where the holes will be there, and in case of cutting seeds. So these seeds are filled in drum in which the small pins are fitted, and while the slow movement the pins are sticking into the holes present in seeds, and these are separated by the brush, so good seed and damaged seed will be get separated. Now we will study about plant protection measures in cotton hybrid seed production. In cotton crop, American bollworm is one of the most dangerous pests. For the control, the BT cotton was developed, about which we will discuss now. BT cotton is a genetically modified organism or genetically modified pest-resistant plant cotton variety which produces an insecticide or toxic substances to combat bollworm. This is a genetically engineered form of natural cotton. The cotton bollworm complex in India includes the old world bollworm or false American bollworm, Helicoverpa armigera, then pink bollworm, Pectinophora gossypiella, spotted bollworm, Aereus vitella, and a spiny bollworm, Aereus insulana. All these belongs to the insect order Lepidoptera. The tobacco caterpillar Spodoptera lituera, also a lepidopteran, which is a sporadic pest on cotton. Although predominantly a defoliator, it can also damage cotton balls and squares when there is a severe outbreak. Among the cotton pest bollworms, Helicoverpa armigera is the most dominant and difficult to control, chiefly due to its widespread insecticide resistance, multivoltine and prolific pattern of breeding, and high polyphagy nature. It is a highly destructive and wasteful feeder in the sense that a single larva can damage many squares and balls. So to combat, the BT cotton was developed. In BT cotton development, the bacteria Bacillus thuringiensis was used. Bacillus thuringiensis is a soil-borne bacterium which is toxic to insect pests and safe for higher animals. It is widely used as a bacterial insecticide. Cry genes from the bacteria determine the action against pests. In India, the first approval to BT cotton were given to 3 hybrids released by Myco Monsanto Biotech, the joint venture between Myco and Monsanto, in 2002. These hybrids contained the BT gene Cry1AC owned by the US firm Monsanto, which licensed the gene to MMB in India. In 2006, the regulator Gene Engineering Approval Committee GEAC also approved 3 other genes constructs MMB Bolgard 2, which stacks Cry1AC and Cry2AB genes, a modified Cry1AC gene developed by IIT Kharagpur in collaboration with JK Seeds, and a fusion Cry1AC and Cry1AB gene which was sourced by Nath Seeds from the Chinese Academy of Agricultural Sciences. Now to study how BT technology works. In case of cotton, where the BT technology has been developed, crop is inoculated with BT gene where the spores and crystalline toxins are inserted. When caterpillar consumes foliage treated with Bt gene, within minutes the toxin binds to a specific receptors in the gut wall and caterpillar stops feeding. Within hours, the gut wall breaks down, allowing spores and normal gut bacteria to enter the body cavity and the toxic dissolves. In 1 to 2 days, the caterpillar dies from septicemia as spores and gut bacteria proliferate and multiply in its blood. So this is the mechanism by which the caterpillars are getting died and it is controlling the pest of bollworm. The second major pest emerged is pink bollworm. Earlier it has also control mechanism in Bt, but now slowly it has developed the resistance and now it is damaging the crops in a larger scale. In case of pink bollworm symptoms, the flowers will be under rosette symptoms and it will enter the balls and it will damage, and the larva colors will be pink color, so it is called as pink bollworm. We can see in the seed they are making holes while entry and eject, and the seed is losing its quality and having the lot of losses to the farmers. So we have to follow the necessary mechanism for pink bollworm control. For control of pink bollworm, in addition to chemical control, pheromone traps are used. A pheromone trap is a type of insect trap that uses pheromones to lure insect. Sex pheromones and aggregating pheromones are the most commonly used. Lure having female fragrance towards which male insect is getting attracted and trapped, and mating is disrupted. Our main is to using pheromone trap is to catch male population of insects and checking the mating with female, by which we can stop the multiplication of insect population, by which there will be control. There are different type of traps which are used. Some are having the plastic tubes under which the insect are getting trapped and they are unable to come outside, and some are sticky pads are also used where the insect are attracting to female lure and they are pasted and sticked to the gummy structure. So different mechanisms are there for controlling the insect population in pheromone traps. Another pest are sucking pests which are causing the damage to the cotton crop. One of them is aphids. For control, we have to use imidacloprid or superconfidor or admire at the rate 5 to 7 ml per tank, and it should be sprayed on frequently basis to control effectively. Other pests are whiteflies, which are also sucking pest insects. So for the control, we have to use the oberane or other chemicals like acephate for the control of whiteflies. Jacids are other sucking pest which is damaging the cotton crop. For the control, we have to use superconfidor or admire or other chemicals for timely control. Thrips are another sucking pest. For control, we have to use reagent or other chemicals available in the market, and it should be sprayed timely for effective control. Mealybug has been emerged as a major cotton pest which is causing the damage to the cotton crop. So for control, we have to use admire or some other effective chemicals available in the market, and it should be sprayed on timely for control. Otherwise, this mealybug is causing lot of damage to the cotton crop which is having the sucking pest category. Aspidoptera insects are the foliage pests which are causing the damage to the cotton crop, and these are feeding on leaves. So for control, we have to use FAME or coragen or other chemicals, and it should be timely controls, otherwise the whole crops can be damaged. After seed production and supply and sampling, the seed should go to quality check, among which the first one is grow out test. It is conducted in field condition where the seed lot samples are shown in field and the all data are captured related to morphological characters and reproductive characteristics. Grow out test is conducted to determine genetic purity of a seed lot wherever it is prerequisite for grant of certificate, and also on the seed lots where a doubt has been arisen about the genetic purity. The grow out test can be complemented by certain related laboratory tests also. The grow out test should be result is more than 90% where the lots will be accepted. If it is below 90%, the lot will be rejected. Second test is germination test. The objective of the germination test is to determine the germination potential of a seed lot. The percent germination is the estimated viability of a seed lot which is tested under specified conditions and within a specified period of time. Each test must consist of 400 seeds which are drawn from the working samples and then randomly divided into 4 replicates of 100 seeds or 8 replicates of 50 seeds. If the germination test is recorded above 75%, the lot will be considered as okay. Third test is ELISA test, which is enzyme linked immune sorbent assays, which are being used extensively for the identification of Bt protein in Bt transgenic crops. A sandwich ELISA test is the most preferable immunoassay for the quantification of Bt protein in transgenic cotton plants, and the ELISA test results should be more than 95%. Now moisture test. The objective of a moisture test is to determine the overall moisture of a seed lot to establish optimal conditions for storage and to maximize the lifespan of the seed. Moisture testing must commence within 24 hours of receipt at the testing facilities, and the standard moisture in cotton seed should be in between 8 to 10%. Then purity analysis. The purity analysis of a seed sample in the seed testing laboratory refers to the determination of the different components of the purity like pure seeds, other crop seeds, wheat seeds and inert matter, etc. And the physical purity should be more than 98%. So these are the major tests which are carried out for the quality checks, and there are another tests also which are conducted for the quality check. So this presentation was all about cotton hybrid seed production technology. Now my presentation ends here. Hope this will be very useful to all of you. I have given here my YouTube channel details, Journey with Vijay Kumar Shrivastav. Having request, please visit the channel, subscribe it and provide your kind and valuable feedback for further improvements in next coming presentations. Thank you.